gave are also really the examples of working together. So I think that's the major message that I want to give. I want to share my story, and hopefully, this will have a better understanding. So, just to share a little bit about me, several years back, almost 3 decades back, I graduated as a medical graduate, and then I had the opportunity to join CCMB, and it has been a fascinating journey. While it was pretty easy for me to get into the shoes of a medical practitioner, but my foray into the genetics and human genetics and really looking at the how the genetics can influence the disease susceptibility or disease management has not been a smooth journey. So now currently I'm no more a clinician for sure, and as I will tell you that I'm really a confused geneticist because of lots and lots of information. And then I realized that if you really want to make any impact on from genetics or genomics to healthcare, then you need to learn epidemiology, which obviously I haven't learned completely. And lastly, all the missing heritability of any of the genetic association is not explained just by genetics. So one needs to look at the epigenetics, which is a fascinating branch that has come up recently. And I'm just trying to get into the epigeneticist mode. Combining all these, I'm trying to utilize, and probably everyone's domain or the dream is to develop that into the public health policy. So, at the first, I would like to acknowledge my lab at CCMB, and this is only the current bunch of people, but I have been privileged to have several hundred people who have actually spent time with me and have allowed me to stand in front of you and sort of deliver this talk. Also, thank my collaborators, and again, I am privileged to have both national and international collaborators. As I go on, I will try to show that how these have been so helpful in getting the information and being able to say that there is something which causes disease susceptibility, can be modified, or can be used as a diagnostic tool. Three just thought these are which I always put. So just I apologize if you find it very naive or very primitive, but just to say that it's the central dogma which we all understand that DNA, a double-stranded molecule, then it forms mRNA through transcription and then protein through translation. The point I'm trying to make is that it's the DNA sequence or the sequence of the nucleotide bases in a particular order, which in fact determines the sequence of amino acids in the protein. So, if there is any change in the nucleotide sequence in the DNA, we would assume that that is going to have a change in the amino acid sequence and thus also influence the protein structure and its function. Of course, it is not always true because we all know that the large section of the genome is intronic and that acts as a buffer for these mutations and their effect is absorbed. So what all of us are actually made of the same kind of DNA, and we know that the functional unit which is present on this DNA is called gene, and all of us are made of thousands of genes, which at least form one protein. Form one can form also a number of proteins. But suffice it to say that there are 30 can form 1,000 genes and close to 130 to 150,000 proteins, and they are largely similar, but yet all of us are different. And this is what my earlier group is there. So, what can go wrong? I mean, what is it that we are bothered about when we talk about genome and disease or any phenotype alteration? So, there can be various things that can happen. There can be a deletion, there can be a deletion, or there can be just an incorrect spelling where, say, a nucleotide base is replaced by another. Then subsequently, we can also have situations where genes can be again deleted or duplicated, or they may be damaged because of some exposure. And of course, we also know situations like Down syndrome or Klinefelter syndrome, where a chromosome can be deleted, or an additional chromosome can be there, or there can be damage because of the radioactive exposure or other things. Now, suffice it to say that all these three can be combined in one fact, that this leads to a change in information, and that to change in the proteins produced, either in terms of quantity or in terms of quality, so that the function of this protein is either lost or the function is gained. And any of these situations, and the implication that any change is any such change, any amino acids, changes levels to be altered, then these are more likely to be associated with a phenotype or something which we change.
Sir, your voice is breaking. I think there is a connectivity issue again.
Can you hear me?
Yes, sir. Yes, sir. Now we
Hello.
can hear you, sir.
Okay.
Yes, sir. We
Sorry.
can hear
Yeah.
you, sir.
Okay.
Please go ahead, sir.
So I said, okay. Please interrupt me. If there is anything that's not going well. Also. Okay. Yeah. So
Sure,
as
sir.
I
Sure.
said, so these are the three genotypes at this particular locus, what we call as wild dominant, mutant, wild homozygous, mutant homozygous, and heterozygous. Now, taking clue from here, most of the genomic research which has an implication in disease, again, this is locus they are here with the patient and defines the disease as best as possible, because everything is driven to the phenotype. If your patient categorization is not that well done, then obviously any of the conclusions drawn from genomics is not going to be successful. Then you determine that what is the disease likely to be genetic, how much is the genetic component of the disease, by twin studies or family studies, where it shows that there is a high probability of family members getting affected than the usual probability would be. And then these are various genetic tools that can be applied. One can identify chromosomal regions as a combination of the patients and the normal subjects. And this can be done through various approaches, which I'll cover a little bit in my talk subsequently. And you identify these regions, then these regions may be large.
Sir, we can't hear you, sir.
In the function we provide out, where we can knock out to understand the implications of these mutations. And as I'm sorry, anything understand the sorry. Okay. So and then finally, it's not always that the each of the polymorphism identified has functional implication. We also need to look at how this polymorphism or the genetic changes, they interact with the environment and induce epigenetic changes, so that there is an influence on the disease development, disease prevention, and therapy. And finally, the result of all these genetic epigenetic studies have to work towards taking it back to the patient so that the patient gets benefited. And if I use a very strong comment that all diseases are genetic, probably a few years back I would have been booed. But today I can say this with reasonable confidence that all diseases are genetic. We can have single gene disorders like beta thalassemia, where we have mutations in beta globin gene. Then we have polygenic disorders like Alzheimer's disease, where we know that amyloid precursor protein is the major gene, but there are many other genes like presenilins or apolipoprotein E variants in whom can also influence the risk of developing Alzheimer's disease. Then we have complex genetic disorders like diabetes, bronchial asthma, hypertension, which essentially are a result of interaction between a large number of genes, but also are influenced by environmental factors like nutrition, physical activity, or lifestyle. And lastly, infectious disease also has genetic determinants both at the host as well as the organismal level. So overall, these different larger categories of diseases, each of them have a genetic component. Now, to put things in a perspective, it can be said that there are largely two categories. One is monogenic and one is a complex genetic disorder. In a monogenic disorder, as this cartoon shows, that you have a particular gene mutations in which cause the particular phenotype. And because this is one gene, various variants, so there is a specific inheritance pattern. It can be dominant, recessive, or X-linked recessive. But again, because of this nature, the genetic risk in the population is similar, and also the genetic risk in different families is also going to be similar, because the presence of mutation indicates a disease and absence means an individual will be a healthy individual. Now, it was several years back that I joined CCMB, and I started with the simplistic part because being a doctor, I wanted to take this whole thing back to the patient. And it is always a good idea to also look at the literature and then use that for the benefit of the patients. So, this is what my stage was when I had joined 25 years back, and clearly the situation has changed quite a bit. Now, let me just tell you that when I was doing this, how the things were. So, if I'm looking at any of the diseases and the genetics of that disease to be beneficial to the patients, as I said earlier, also that one needs to take a good clinical and family history so that the diagnosis is established, and family history determines whether it is genetic disease, what is the pattern of inheritance, and then one establishes the clinical diagnosis by using various biochemical or radiological parameters. Again, to reiterate that if the clinical phenotype is not very well established, any further studies on genetics is not likely to be useful. And then by using various tools of genetics, identify the genetic defect that is responsible for the disease. And depending on the nature of the inheritance of the disease, screen for carrier status in the recessive disorders, and then finally take all of this information back to the family and identify the carriers and do a pre-pregnancy counseling, so that whenever the lady gets pregnant, then the couple approaches for a counseling where it can be communicated that what is the risk for having a baby with a genetic defect that is running in the family, or even there can be some de novo situations as well. And at the last, one does a prenatal diagnosis, which means collects the amniotic fluid or the chorion villus sample from inside the womb and does genetic testing and tells that whether the baby is likely to be affected or not. The affected babies get aborted, and it's the healthy ones which are allowed for the pregnancy to continue. Now, let me give you an example of sickle cell anemia. I'll not go into much detail because I assume all students would be knowing about it. But suffice it to say that this is a pedigree of an autosomal recessive disease where we find that two individuals are affected and their parents are obligate carriers following the pattern of recessive disease. And you can see that if an affected individual marries a healthy individual, all its kids are going to be heterozygous, and then if the carrier marries an healthy individual, then there are different probabilities. And this is exactly what one can find out by doing a pedigree analysis. So without a massive knowledge or without a massive infrastructure of sequencing or anything, it is easy to identify what is a genetic disease and whether the family can have anybody at risk or not. Now, as I was mentioning, that it is the sequence of the bases that determines the protein function, protein structure. So there is an A to T transversion in codon 6 of beta globin gene, And as you would notice here that this changes the amino acid glutamic acid is replaced by valine. And because of that, a sticky patch is generated on the surface of erythrocytes and they become sticky and also become sickle shaped. So, this is what is the phenotype of sickle cell anemia. Now, once I have the family history to further substantiate the diagnosis, one can do various biochemical tests, like, say, for sickle cell anemia, one can do hemoglobin electrophoresis. And depending upon the presence of bands, like the sickles band will not be present in a normal individual. And you would see that for an affected person, there is a higher sickle cell hemoglobin amount compared to heterozygous who has equal amount of sickle hemoglobin as well as the adult hemoglobin. Now, then one can use different genetic tests. And the point of showing this particular slide is that it can be as simple as a PCRRFLP, which means that you amplify the fragment which is harboring this mutation and then use a specific restriction enzyme which creates a site in the presence or absence, in the presence and absence of which site, which mutation the restriction enzyme would cut or not cut, and that will give us a differential.
Sir, audio connectivity lost again, sir. Sir, can you hear me? Sir, audio connectivity lost, sir. Sir, audio connectivity lost, sir. Can you hear me, sir?
Yeah, okay.
Yes, sir. Yes, sir.
Can I go ahead?
Now,
Okay.
please go ahead, sir.
Okay, so I just mentioned that once you have the genetic defect identified in the family that is responsible for the disease, then you can just take a prenatal sample from a pregnant woman's womb, and these can be the nature of samples, isolate the DNA, and conduct the same genetic test as you would have done earlier to establish the genetic status. Look for maternal contamination because these are blind methods of ultrasound mediated sampling. Hence, one can look at the maternal contamination and then finally have an idea whether the baby in the womb is affected or unaffected. And then there has to be genetic counseling, which, as you all are aware, I'm sure you all are aware that for dominant disorders there is a 50-50 chance. There is a autosomal recessive disorder, there is a 1 chance in 4 for a baby to be affected, and for X-linked recessive again it's 25%. But because it affects only the male, so out of the males it is 50%, but overall it is 25%. Now, clearly, with this kind of an information, it is easy for the couple to take a decision whether to continue the pregnancy or not. And this is the way one would do counseling. They would inform the nature and consequences of the disorder, then probability of developing and then transmitting it to the future generation, because that's what the parents are largely concerned about. And then, is there any immediate treatment available just after the birth? There are diseases that actually need some amino acid supplementation, like phenylketonuria, and that the disease can actually be taken care if a modified diet is given in the early life. Then prenatal diagnosis, pregnancy monitoring, and last but not the least is to support the parent's decision. This is important because Many of the couples or the families, they are not completely aware, but it's our duty to communicate to them the proper risk and the future development so that they can take a call accordingly. It is now known that a family which has Down syndrome kids, they actually don't want to abort the children. So in US, Down syndrome is not really made as something mandatory for which the abortion can happen. So just for this much part, for a single gene disorder, life seems to be pretty straightforward. You diagnose the disease, you identify the genetic defect, you look for the pedigree chart, identify the carriers, caution them that these are the possibilities, provide them the approaches and the solutions, and then one can help the family in having a healthy baby. Now, this sounded pretty simple, but slowly, over time, it came to be known that there is a variable clinical severity of sickle cell anemia. So you would imagine that if there is one mutation which is causing a change in particular amino acid, and if there is a influence on the protein function structure, then the disease phenotype would be simple. I mean, that's the notion with which we go, that genetics drives the phenotype. But there has been variable clinical severity in different parts of the world for sickle cell anemia. Like Africans have very severe phenotype and Indians have relatively mild phenotype. And on investigation, it was found that there can be co-inheritance of alpha thalassemia, because alpha thalassemia also leads to high fetal hemoglobin level, and this fetal hemoglobin level is known to influence the sickling of the erythrocytes. And then it's shifted towards looking at whether there are genetic regulation of fetal hemoglobin levels, and that's where a lot of studies have been conducted. Last but not the least is that initially it was only thought with only one mutation the sickle cell phenotype can occur, but actually subsequently it came to be known that there are various mutations in the beta globin gene cluster, which has not only beta but alpha, gamma, all those which actually form the hemoglobin at different stages of life. And in fact, that is what is called the haplotype background against which the beta globin gene has mutated. Now, this also has implication for hydroxyurea therapy for sickle cell anemia, because this particular one mutation which influences the fetal hemoglobin level. So this was a study which we conducted long time back on the coastal region of Andhra, where we looked at two tribal populations which were specifically suffering from sickle cell anemia due to high degree of consanguinity. And what I was mentioning, that these are different variations which are present in different globin genes like epsilon, G gamma, A gamma. And what we find is there is a specific pattern in all these haplotype backgrounds. Now, this Benin, Bantu, Cameroon, Senegal, they are all African haplotype. But if you see the Indian haplotype, you would see that it is at this particular polymorphism. It is not so in all the African haplotype, but it's present in Arab, Indian, and Senegal. And this was subsequently known to be associated with fetal hemoglobin levels. And in fact, we found that the Arab Indians, they had fetal hemoglobin up to 30 to 40%, which is in contrast to the Africans who had only 5 to 10% fetal hemoglobin. So there was a clear cut influence of the variants in the globin gene cluster other than the beta globin gene mutation which causes sickle cell anemia. So this drove 2 points. One is that only one mutation will not decide the final phenotype, and which means that you have to look around the gene cluster. And second thing is that is likely a causal mechanism by which these variants will influence an intermediate trait like fetal hemoglobin in this particular case. So this was sort of the first indication that genetic modifiers might play an important role in monogenic diseases, because people thought that the story of sickle cell anemia is over. Everything is done, the mutation is identified. But no, friends, as technology went on, there are now genome wide association studies which look at millions and millions of SNPs. And just with that approach, today we know that there are about 38 genes which regulate the fetal hemoglobin levels. Remember, I talked to you only about one polymorphism in the A gamma globin gene, but now we know that 38 genes are there. And there are specifically three genes, these three genes, which account for 20% variation in fetal hemoglobin levels. So this clearly means that multiple genes may interact with each other to decide the fetal hemoglobin level. And despite all these efforts, we only 20% heritability in fetal hemoglobin level known, which means that there are still possibilities of either finding genetic variants or there could be other mechanisms by which the fetal hemoglobin levels might be altered. And this is possible probably through epigenetic regulation, which doesn't change, which doesn't work through the change in the sequence of the nucleotides, but it does modify and put certain modifications on these bases which allow the gene and the gene expression to be regulated differently. And most importantly, many of these genes that show association with fetal hemoglobin also are associated with complications of sickle cell anemia. To summarize, what we are looking at is in sickle cell anemia, which is a genetic disorder, the levels of fetal hemoglobin can influence the severity and the complications. And further, we also know now that there are many genetic variants in these genes which can influence the fetal hemoglobin level. So the implications is that even when the child or the baby or the individual is diagnosed with sickle cell anemia with a specific mutation, yet there are still possibility of doing genetic testing which will help in the predictive testing of an individual having further complications as an outcome of fetal hemoglobin levels. So, friends, that brings us to monogenic versus complex genetic disorders. You saw this picture where I told you that it's one gene, mutations in that gene causing the disease. There is a specific inheritance pattern, and the risk, either in the population or in different families, is likely to be same. But contrast it with the complex disease. As I mentioned to you, can you hear me?
Yes, sir, we can hear you.
Okay.
Yes, sir.
So, in contrast to the monogenic disorders, you would see that there are n number of genes as shown over here, and each of these genes will have their own inheritance patterns, which is why the inheritance pattern for the disease would be complex. And finally, as there is an influence of environment, lifestyle, as I mentioned earlier, hence there will be a different gene environment interaction, and hence the risk of the disease in different populations will be different, and also in the say in the different families it will be different, because each of the families may have different risk due to the genetic factors. So as I mentioned to you that this is what I was when I started, and this is what I am today in 25 years time. So this really tells us that how the knowledge about the genes, the technologies, and their application in these genetic diagnostics and testing has helped us understand this even better. Let's move to complex disorders. This cartoon I have taken from one of the presentations, and is something basic which we need to keep in mind if we want to design a research which will help us understand the genetic basis and also help us to take this back to the patients, which is what was the chart that I showed you. So if we think about cardiovascular disease, that is really a complex disease. Now you can see that if I talk about the disease risk for a complex disease, there are going to be various genetic risk factors, various protective factors, and as I said, that complex disease is also influenced by environmental factors. So there are environmental risk factors and environmental protective factors. So finally, finally, the disease status is determined by an interaction between these risks and the protective factors or the risk factors of the environment. Now you know that if you are carrying some of the genetic variants which predict risk for, say, cardiovascular disease, They can actually influence your age of onset. There can be early age of onset. The disease can be severe. The clinical symptoms may be extremely variable. But then all many of these risks can be attenuated by taking a healthy diet, by doing exercise and modifying your lifestyle. Like you all know that environmental risk factor means if you carry high lipid levels, then by doing exercise or taking a lipid free diet, or not smoking or not taking alcohol, one can actually alleviate the risk that is posed by these genetic variants. And that is the excitement of complex diseases, because genetic variants can't be changed so easily. We have already heard Professor Padmanabhan, there are many techniques, many technologies, but most of them are largely, ornamental stuff. They have been shown as proof of principle, but it's not so easy in real practice. And second point is that because this is a complex disorder, so there are large number of genetic variants which will be influencing the risk. Hence, how many variants can one change? And just now we are struggling with just one genetic change. To make one genetic change, such as sickle cell anemia, this will be even more impractical and may be fraught with more and more dangers. So, this lower cartoon indicates that further complication of these genetic factors is that if we look at the environmental risk factors, like say your own constitution in terms of ACE level, blood pressure, HDL, triglycerides, you know that if these are high, then they make an individual at a higher risk of developing disease. And you would notice that while angiotensin converting enzyme levels are predicted only by ACE, but you would see that HDL or triglyceride, its levels are determined by so many genes, and hence it is extremely difficult to design a simple genetic testing or a treatment option. Now, I work on diabetes, and I would like to share my story with diabetes. And I would again like to acknowledge the contribution of my collaborators and my students and my project staff in helping us achieve something. So you know that diabetes is a global emergency. Now we started with sickle cell anemia, and we could find that the prevalence of this disease is not very high, and so, even if we are doing something on genetics, we are helping a small section of the community. But we also wanted to spread it to a common disease which affects a larger section. And you might be knowing that diabetes, India is literally fighting with China to reach and become the diabetes capital of the world. And currently the prevalence, this is a little old slide, but currently the prevalence ranges between 15 to 20%, which means that if there are 100 people sitting in this WebEx, close to 20-25 are actually diabetic. And it is also known that diabetes has strong genetic determinants. If you can remember my first slide where I said that how do we determine that a disease is genetic? There is a high rate of concordance in monozygotic twin, and there is an evidence of familial aggregation, which means that the possibility that the child of both diabetic parents is going to be pretty high compared to an isolated parent or normal couple. And then we also know that there are intermediate traits like obesity, insulin resistance, insulin secretory function, which are risk factor for development of diabetes, and they all are under strong genetic regulation. Just one sec, I'll have a gulp of water.
Yes, sir. No issues. Please go ahead.
Okay, so when we started working on this, particularly with the notion that it's the genotype that drives the phenotype, when we looked at the phenotype of diabetes in Indians, what we found that in contrast to Europeans and Americans, we have a very different phenotype. So the Indians developed diabetes at least 10 years earlier than Europeans, and they develop diabetes even though they are not obese. So even if they have lower BMI, they develop diabetes. And then mid-arm circumference, which is a measure of the of the fat mass, that also, if it is lower by 20%, still the diabetes risk is 15 to 20%. What we also found that the Indians have more of obesity and insulin resistance. As I am showing over here, that the Indians are 1.5 times more insulin resistant than the Europeans, and in fact, their fat percentage is higher than 30% or so. I'm sure you don't believe me, but let me assure you that Indians are obese in a very different way. So this was the era between 2006 and 7, and genetics and genetic technologies were at its highest. We had the evidence that the phenotype is different, and so we started to use the hammer of genetic studies with a hope that genetic studies will explain everything. And we again applied this whole notion that I have mentioned to you earlier, and we wanted to look at the polymorphisms and then look at whether they are in the genes which influence the intermediate traits of diabetes, and then identify how do they interact with each other. Now, diabetes has been really a big, geneticist nightmare. It has been worked on for several years. I'm just trying to tell you here that what are the approaches that can be taken for identifying genes related to a complex phenotype. You can replace type 2 diabetes with any complex phenotype like hypertension, cardiovascular disorder, Parkinsonism, anything, but the approach is largely the same. So what has been usual approach earlier, that you take the phenotype carefully and identify that what are the clinical features, and based on that decide the gene that could be associated or be responsible for the disease manifestation. For example, you know that hemophilia A is a disease which is where factor 8 levels are low, and hence the immediate candidate gene is factor 8 gene. And in fact, that is what was done using candidate gene approach, and a number of mutations, close to 250 mutations, have been identified in factor 8 gene. Similarly, for diabetes, people started taking a candidate gene approach, but it was not so easy because, like hemophilia, it wasn't a single abnormality that would define type 2 diabetes. Actually, type 2 diabetes is a syndrome which is characterized by high glucose levels and high insulin resistance, low insulin secretory function. All these things combined give it a diagnosis of type 2 diabetes. So they looked at pancreatic beta cell function, they looked at insulin action, glucose metabolism, and in fact, there were only largely two genes which came up prior to 2007. And then came the approach a little bit advanced of linkage studies, where, as I mentioned earlier, that one looks at clear cut diabetes and clear cut normal individual and does a linkage, which means that you are comparing the large sections of the chromosome between cases and controls, and whatever regions show statistically significant difference, those regions are then taken and are then dissected to identify specific genes. There also it's been very disappointing, but there was one gene TCF7L2 which was identified, and this has been substantiated in subsequent studies. So the point I'm trying to make it that while phenotype helps you in many things, but genetic technologies advancement has also helped in identifying new genes, which means that there are genome-wide association studies which allow you to look at the whole genome, and you need to have thousands of cases, thousands of controls, and of course, then you will be able to identify common genetic variants. Now, it is important to appreciate that the common genetic variants will be some which will be helpful in predicting the risk of complex disease like type 2 diabetes. Now, I just told you that about diabetes, type 2 diabetes, but again, it's not as simple as the phenotype will tell you that this is type 2 diabetes. Now, take a look at this picture. This is the diabetes in young adults. So, imagine an individual aged 30 years who comes to the clinic and shows symptoms of high glucose levels. If the individual is pretty young, this is what is type 1 diabetes, which peaks at between 15 to 20 years. Then here is an individual which will be type 2 diabetes, which will usually peak between 40 to 45 years. Then we have this individual who is actually MODY, which is maturity onset diabetes of the young. It is an autosomal dominant disease, almost a monogenic condition, which is caused by mutations in only one particular gene. Now, this also occurs between 15 to 25 years or so. So, the point I'm trying to make is that there can be diabetics who can be presenting at different ages. But in India, there are individuals who develop disease at least 10 to 15 earlier, which means that even at the age of 25 to 30, there is a possibility that an individual could be type 2 diabetes. So while the clinical picture, the age of onset helps you get to the status of identifying or having a provisional diagnosis, but clearly genetics can help further. How can it help further? I'm just giving you two or three examples to drive the point that how with these genomic studies and genetic studies, things can be taken to the benefit of the patients and the population. Now, as I mentioned to you that maturity onset diabetes of the young is actually an autosomal dominant disease, and this disease is pretty severe. There is a early diagnosis of diabetes less than 25 years, and it is caused by single gene defect. Now, by describing this MODY and type 2 diabetes, I'm trying to bring the perspective that even though you may have a complex disease or a complex phenotype, it is always good to go back and look at the monogenic entities. As I told you, the example of sickle cell anemia and then the hemoglobin, fetal hemoglobin, which can cause various other diseases. Similar is the situation over here. What we say is that all these family, you would see that there are so many individuals even as early as age of 9. So when you look at the genetic causes of MODY, there is again further diversity. There are some 15% which are caused by mutation of glucokinase, which you know is the rate limiting step. And then there are close to three-fourths which is caused because of different transcription factors, the HNF group of factors, and 11% for which still we don't know what the gene could be. So again, the message is that even though we think this is a MODY, but there is an evidence of genetic heterogeneity, which means that the same phenotype can be caused by mutations in number of genes. Now, I want to give you the example as I showed you that there can be glucokinase 15%, 75% can be because of this transcription factor mutation. Now, you on the x-axis is the age of an individual and y-axis is the glucose level. If you take two individuals of MODY, one with glucokinase, one with transcription factor mutation, you would see that the normal individual, the glucose goes like this till the age of 80, it remains as stable level. But if there is a glucokinase mutation, the baby is born with a higher levels and it continues to maintain the levels at a steady level even at the age of 80, which is marginally higher but never reaches the diabetic range. Now, if you see a transcription factor mutation individual, the individual is born normal, but then pretty early, within 15 to 20 years of age, the glucose levels start shooting high. And so, point is that the glucokinase mutations are milder and transcription factor mutations are severe. Now, this you should imagine in the background of an individual presenting with diabetes at the age of 15 to 20 years. So clearly genetic testing has a role to play because you can use the treatment for specifically glucokinase. You may not need to do any treatment because they are only having a little high levels of glucose and they can be easily managed by the diet itself. Whereas if you go to transcription factor mutations, then you would specifically be needing insulin. At least one third of the individuals would need insulin. And because there is a poor control of diabetes, the complications are also quite frequent. Now, one example of doing genetic studies even on a monogenic disorder and what benefit it can have. Now, further, in terms of the management of the disease, you know that for diabetics, there is insulin or there are oral hypoglycemic drugs, and there are two kind of drugs. one is sulfonylureas, and one is biguanides. Now, on the x-axis is the years since an individual is diagnosed as diabetic, and on y-axis is the glycosylated hemoglobin, which is the measure of glucose over a period of about 3 to 4 months. Now, if you would see that this individual, and 6 is the cutoff for glycosylated hemoglobin, you would notice that this individual is normal. He is specifically on glibenclamide. So he is continuing on that treatment from 8 years, 10 years, 11 years. And then this is a trial in which glibenclamide was stopped and metformin was started. Now you see that just after the stoppage of glibenclamide, the metformin couldn't control, and this went on increasing. Again, at within 2 years' time, when metformin was stopped and glibenclamide was given, again you see the glycosylated hemoglobin level started going down. And this is the heterogeneity with reference to the transcription factors. So I showed you that there are at least 12 transcription factors, mutations in whom can cause diabetes. But there also it is important to identify which transcription factor, because mutations, individuals having mutations in HNF1 alpha are sensitive to a specific treatment. So friends, I think I have been able to communicate to you that why genetic testing is important and why it is also important in complex diseases which are combination of traits, because even if you identify one, two, or any genetic variant, there can be implications in diagnostics, in treatment, or in follow-up for looking at various complications. Let us just move a little bit further. There have been lots of technical advances in genetic studies. As you know that monogenic versus complex is a big dilemma, and you need more technological advances for dissecting the basis of complex disease. When we started in early 80s, it was only one variant. We used to use gel-based method, as I showed you, and then subsequently people started looking at number of SNPs in a particular gene, and then it started with multiple variants in multiple genes where 90, 100 variants could be analyzed, and then with technology advances, there were more than million chips, which million SNPs in a chip, which came around 2005, 2006. And then now we know that we can do exome sequencing, whole genome sequencing, and everything. Now, is it that more is always better? Or would you think that when you were doing targeted or directed genetic studies, you were better off? Now, the answer lies somewhere in the middle. If you have more data, that doesn't mean that you will be able to get all the information. And this is because whenever you do multiple testing, whenever you have large amount of data, there are associations which can just come by probability itself. So what you do is you do the experiment, and then you look at a high statistical significance, which is not p level of 0.05 but 10 to the power of minus 8. So if on association analysis using regression or using chi-square, if you find any variant which passes 10 to the power minus 8, then you put that as statistically significant. And then further to rule out probability, you do again the genotyping for another round of patients and normal subjects, and then see that which of them again pass the high statistical significance. This is all to rule out the probability that by mistake or by false way that a genetic variant has come to be associated. Now, the next concept is that you start with one study and you find something, you may be that you have found certain genetic variants which are associated, but there are many factors which can influence the strength of association. Now here is an example. PPAR gamma, which is a protective gene for type 2 diabetes, you would see that these are different studies that were conducted. So this is the graph which shows the odds ratio. Now this is one. Anybody on this side which is affected, and this side they are susceptible. Now you see with small studies like 400 individuals, 350, 370, there is a high variability in odds ratio. You see, this individual shows zero point 0.3, whereas this shows about zero point 0.9. Now, the important message here is that if you do any such studies with small sample sizes, it's very difficult to find because your study may not be adequately powered. Now, you see here a big study which was on 3,000 samples. It showed that the odds ratio is zero point 0.8, which means an individual carrying the risk allele is protected by 20%. And you see that if you combine all the data by meta-analysis, then you see the similar results. So the point I'm mentioning is that while you do any study on complex genetic disease, you need large number of subjects, you need large number of or a genetic technology which gives you massive genomic data. Then only you will be able to have results with confidence. Needless to say that you need good bioinformaticians and biostatisticians, which is a very good career at this point of time because they are required everywhere. So this is what is the principle of association testing, as I showed you earlier. So you take cases, you take controls, and then you run them on chips which have millions of the steps. What is an association analysis using regression? And what you find is called, this is called as a Manhattan plot. In x-axis is the chromosome, and on y-axis is the p-value. And you would notice that there are many of these dots which are passing above this particular line. So each of these dots represent a particular variant, which is a SNP, and this line is the p-value cutoff of 10 to the power minus 7 or minus 8. So this is the way that the association testing can be conducted. And as I showed you that you don't rely on only one study, you rather do several studies on different cohorts and then do a meta-analysis, and then you are able to predict conclusively that what is the odds ratio of developing the disease. This is just to show an example of the results that we did. And this is, you see the Manhattan plot, and you would see that many of these gene variants have come up. And this is something in 2007. Now, okay, you found some, at this point of time, there were 10 genes which were predicting the risk of diabetes. Now, can it be used for any prediction, And what will be the role of environment? That's the question with which we had started. So, what we did was that rather than looking at an individual with a particular variant, what we created was a genetic risk score, which means that we combined all these 10 genes. In fact, here it is 33 genes. We combined them and created the combined genetic risk score as a proof of an individual having all these genetic variants. If you see over here, the individual who was 39 7 times higher risk of developing diabetes than an individual who is having only 17 risk alleles, which clearly means that the genetic contribution leads to a higher risk to an individual, and that's is a genetic disease. But then, when we try to look at how the environment also influences the risk in the background of this genetic risk, we did a ROC curve analysis, which is looking at area of the curve. Again, I'm not going to explain much, but you would see that if we use the genetic risk score on these 36 genes, we find that the area under the curve is only 0.634. When we look at the conventional risk factors like age, obesity, insulin secretion, insulin resistance, we find that the curve is large, it's having a huge area which is close to 0.94. Now we wanted to see that is there any addition of adding genetic risk score to conventional risk score, and we found that the combined the area under curve only increases marginally, only 2%. So this overall indicates to us that while genetic risk is important, but these environmental factors, they are equally important, and there is a possibility that these environmental risk factor might be modulating the risk which is contributed by this by these genes. When we look, so to take this further, we divided these 100 odd genes into different categories, which are known as the intermediate traits. Like, that high glycosylated hemoglobin is a feature of diabetes. So we find that there are so many genes. Then for type 2 diabetes, so many genes. Postprandial glucose, so many genes. And we find that there are many genes which are overlapping with each of them. And this clearly suggested that each of these genes that we are talking about, they have influence on the intermediate traits of diabetes. And that is why these conventional risk factors effect is also mediated through many of these genes. And that was quite exciting because it would mean that if we find a genetic association, there is always a possibility that this whole risk can be modified by the environmental factors, because we can always take care of fasting glucose, we can always take care of glycosylated hemoglobin, and that is what is the excitement. Second part, which is also important, and why the genetics may not be the bane of everything, is because there are different ethnicities with their own genetic constitution. As I told you that Indians have a very different phenotype, and clearly as an ethnicity, the Indians will also have a different genetic constitution. We hypothesize that, well, because Indians have a different ethnicity, because the Indians have a very different phenotype of diabetes, it could possibly be that different genes or different risk genes could be predicting the risk of diabetes in Indians, and that they could be different. Now, what we found, again, to cut a very long story short, we did find some differences, but they were not significant overall differences, which means that largely all the genes that are predicting risk of diabetes in Caucasians also does it in Indians. But importantly, some of the genes do not. So, for example, this is an FTO gene, which was shown to be associated with diabetes very strongly in Europeans. And they also showed that the FTO actually increases the BMI, which is a measure of obesity, and then it influences the risk of diabetes in Europeans. So, if you control for BMI, then the risk of diabetes is completely taken away through FTO. When we analyze the FTO, what we found that FTO associates with BMI. FTO also associates with type 2 diabetes. But even if we control for BMI, the risk of type 2 diabetes is not gone, which would mean that the association of FTO with type 2 diabetes in Europeans is through obesity, but in Indians, it is both way. So the FTO influences obesity as well as diabetes, and this risk could be very different in Indians. And actually, at that time, we thought that there could be a differential regulation of FTO which may be responsible for this difference. So again, to build upon my point that these are three studies which came from on South Asians and Indians, and you would see that none of them have any influence on the diabetes risk. So if I have to summarize the whole research of type 2 diabetes using different technologies up to GWAS, which was the phase in 2016, today there are on that time there were close to 100 variants which were identified for risk of diabetes, and actually those 100 loci will still explain only 12% of overall genetic contribution to type 2 diabetes, which means that if I am carrying the risk allele at all 100 loci and you are not, then I am only at a 12% higher risk of diabetes. Does that sound exciting? Well, clearly, all this effort doesn't really take to that stage where you only tell an individual that, well, you are at a 10% higher risk of developing diabetes if you undergo this kind of test. Now, clearly, the things didn't stop over there, and clearly, there was a review in Nature which said that it still continues to be a geneticist's nightmare. Now, can we do from here, or is it that the story is closed for diabetes? I'll skip this slide because this will take a lot of time. So, Now, as I showed you that there are large number of chips, there are large number of exome studies, large number of whole genome studies, and it is possible to have a number of variants. Now, the proof of the principle for a polygenic risk score came by studies on cardiovascular disease. You already heard from me about genetic risk score. Polygenic risk score is just one step ahead in the sense that it takes larger and larger number of genes, and the technology allows one to do it, particularly look at the effect of each gene on another one. Now, this is sorry for this busy slide, but you take me on my words that from various studies, the scientists identify 53 significant variants which predict a risk of coronary artery heart disease or stroke. What they did was they analyzed these 53 SNPs, and they found that if this particular prediction is applied to those who have a high risk score because of their habits, then they can prevent additional event of heart disease or stroke. Going further on this, now they have moved further and they have looked at close to 50,000 SNPs across the whole genome. An important part is that each of them are not statistically significant by themselves. But what they do is that when they increase this SNP number to 50,000 or so, then they also identify the risk much better so that a proper prediction of lifestyle modification or treatment can be provided. Now, I would like to take it a little bit further, which I'm sure is going to be a success. When you develop this polygenic risk scores, as I mentioned, that you need large number of subjects, you need large number of variants, and you need individuals who represent different ethnicities. What is the future? Uh, Future people are talking about genetic sphygmomanometer, which means that if you have the whole genome sequence or say a large number of variants, then what one can do is take that whole thing and try to find out the risk variants which predict different traits. Okay. Now, the more better example is over here. So suppose if I find a particular variant which influences BMI, or say 50 variants which influences BMI, in that case, because I know that high BMI is a risk factor for type 2 diabetes, cardiovascular disease, breast cancer, and multiple dysplastic disease, then I can use that 100 SNP risk score, which helps me to categorize individuals into high BMI. Then I can counsel the individual that because of these risk factors showing higher BMI, you can be at risk of these four particular diseases, and hence you need to be careful. Similar thing can be done on various treatment options, as I showed that statins, aspirin, they are all associated with cardiovascular and cancer. So clearly, it should be possible if there are huge amount of genetic data available, but I must caution over here that the previous slide, as I mentioned, that there has to be a very clear phenotypic information, clinical information on individuals. This has to be done on large number of individuals, and lastly, this large number of individuals should be having a ethnicity mix. So the last part of mine, which will take maybe 2 3 minutes more, I'm almost close to my time. So now we know that we can use different approaches. So till now I had told you about about the common disease, a common variant, common disease hypothesis. Now clearly you heard that these common variants only explain a small proportion of the genetic risk or heritability. So now with whole exome sequencing, targeted sequencing, and whole genome sequencing, people have been hypothesizing that there could be rare variants which will have a large effect size, and that is why people have gone on to these approaches. Now, whole exome sequencing, as the term determines, that you look at only the protein coding regions, and actually it can facilitate the discovery of disease causing mutations. Now, this is only 1% of the genome, and clearly, if you have a very clear cut phenotype which shows a monogenic kind of pattern, then approach is extremely useful. Second is targeted sequencing, which means that if your phenotype is very clear, like say for example, if you have a muscle disease, muscle dystrophy, but you are not able to diagnose muscular dystrophy with complete confidence because that is highly variable. There are at least 33 types of muscular dystrophy. Then in that situation, rather than going for exome sequencing, which is fairly expensive, one can just take those specific genes and do form a panel and do a sequencing of those regions and then identify the mutation. And last but not the least is whole genome sequencing, which, I mean, suffice it to say that it's massive, it gives extensive throughput, and analysis is pretty complicated. So just to give you an example, sorry again for this busy slide, there is a case of how gene sequencing very early on had saved a particular twin. So just suffice it to say that the individuals had a particular phenotype, both of them, and finally looking at their phenotype, the exome sequencing and other sequencings done, and they were diagnosed as Segawa's dystonia, which is a rare disorder. And it was known that in this disorder, which is because of the problem with the with the just one sec. Hello. Yes. Oh, I'm sorry. I thought you were calling. Okay. So Segawa's dystonia, where there is an issue of dopamine metabolism, and there is a drug L-Dopa, which would actually which was used, and as you would see that these were the kids at the age of 5, and they were treated with L-Dopa, and they could reach this stage, and they could do everything that any normal individual would be able to do. So this is the power of genetic testing. So the message is that any type of genetic testing, if it is done in the background of a clear phenotype, a clear understanding of the genes which are likely to be responsible, it is possible to help make a difference in the lives of different individuals. We also have a PCSK9 story. Again, I'm not going to go into the details of this, but suffice it to say that even for complex diseases like cardiovascular diseases, if you have a phenotype for which you identify a genetic variant, it's possible. So in this particular situation, there were 20. Now we know that statins can reduce the cholesterol levels, but then there were 20% patients who could not tolerate the statin side effect, and actually they were not able to control the cholesterol levels. So from exome study and genome study, they found out that there are variants in gene called PCSK9, and then now this is almost gone into clinical trial, and they are now being used to help individuals who are not responsive to statins. Okay, so now I leave you people with this question that can these genetic variants associated with complex diseases be used for predictive medicine? I showed you some of the example, but how is the hope? Now the fact remains that genetic is fixed at birth, and clearly, as I said, that this cannot be changed. There are multiple genes, there are genetic heterogeneity leading to the same phenotype. Then there are multiple variants. I gave you example of 50,000, each of which have small effect size, and they still predict some 10-20% of the risk. Then there is pleiotropy, which means that there are environmental factors which might lead to similar phenotypes. And of course, we have an evidence of gene environment interaction. So looking backwards, is it an ideal situation to identify the genes which create the risk for a disease? Well, you really need to look at prospective studies, which means that you recruit individuals in an early stage and then try to identify over a period of time how would they develop the disease. And this is the last area that I want to show, that this is something which is known as developmental origins of health and disease, which means that this goes on the notion that the risk of these complex diseases like diabetes, cardiovascular, that is fixed early on. And this is from the notion that this is called Barker's hypothesis. This is with the notion that the maternal nutrition determines the fetal growth, determines the birth size, determines then it determines the development of the child in the same way. And it is known that babies who are born low birth weight, they have factor. So what I'm trying to say is that, sorry, I'm sorry. When we look at a comparison of an European baby with an Indian baby, that India is the diabetes capital and also the capital of low birth weight. So what we found that the Indian baby with a 20 high, something like 27%, 21%, the European baby is 9%. The obesity, which is largely around the which is called the central obesity, that is very prevalent in Indians and it is largely present at birth. So we hypothesize that if we want to do anything as preventive as possible, we probably have to work at a stage where the disease risk can be minimized before the
Voice is breaking, sir.
this particular risk, and hence by doing a B12 and folate supplementation, this can happen. Hello, am I done? I'm very high.
No, sir. The voice was breaking, so you can take 5 minutes more, sir. No issues.
Okay, 3 minutes only.
Yes, sir.
Audio problem, Chandak.
Sorry.
I said there is an audio problem. That's what. Yeah.
Okay, that's fine. Yeah, okay. So just to summarize, I tried to show that if there are women who have a low B12 and high folate levels, then their kids are born with low birth weight. And this is such an interesting observation because we know that the kids who are low birth weight, they finally are at a higher risk of developing cardiovascular disease or type 2 diabetes. What we did was we gave B12 to these people who were having low B12 and high homocysteine, and we found that actually their levels do increase and homocysteine levels drop. Now, this gave us an indication that probably this whole genetic risk is getting modified because of this micronutrient intervention. And if we look at the B12 and folate, they are in the pathway of one carbon metabolism, which decides the methylation potential of the cell. And we know that DNA methylation is one of the important important epigenetic mechanisms which regulate the gene expression. So a small study which we did before and after what we found that, so this is the study design as I showed, and what we found that the levels of key type 2 diabetes genes were significantly altered by B12 supplementation. To take just one more minute, that which means that the kids who were supplemented with B12, they had a differential methylation of these four genes which are known to be the major type 2 diabetes risk, which suggests that there is a possibility of regulating the methylation levels of key type 2 diabetes genes by giving B12 supplementation. Now, we did this in children who were between 9 to 11 years of age. So, we can't exactly say that whether this is possible to use it in adults also, but there are some evidence coming up that with B12, there is a reduction in the risk of type 2 diabetes and complication. So, this is the overall summary that I'm trying to say, that while the genetic risk stays, and these are all the genes which are associated with risk of diabetes, But if B12 supplementation is done, then the methylation potential is altered, and there is differential methylation of a microRNA, there is differential methylation of these particular genes, and they actually influence obesity, insulin resistance, because FTO and TCF7L2, you might remember, are associated with these two intermediate traits. And our hypothesis, which we had put when we found FTO not to be associated through BMI to the risk of obesity, also came true. So the dogma then, from central dogma, or using the approach of central dogma, we now are able to say that, well, type 2 diabetes is a complex disorder. There is genetic susceptibility. There are precipitating factors like obesity, diet, and others. But we bring another component, which is called developmental origin of health and disease, which means that if we can control the intrauterine environment and we can define the baby's health at birth, then probably we'll have a healthy individual. This is what clearly had provided that all common diseases are going polycystic. We don't know how, we don't, but it's taken still took 100 years. So it was in 2008 only that it could be shown. And then this actually provides all the genetic technologies and the environmental risk interaction. It provides insight into shared etiology between conditions. So there are promising application in stratified medicine, which means that you can develop personalized medicine, you can develop population-based medicine, and this should be truth in few years to come. Of course, there are challenges and potential for causal inference, because whatever variants we pick up, they are only by association analysis. However, I'm sure that with analytical methods and cell biology methods, it will become possible to understand the causality of these variants. And of course, there are challenges and potential for individual prediction, because each individual is not as simple as we think it to be. So thank you very much. I hope that many of you people will take this path and be ready to age gracefully like this. Thank you very much. I want to acknowledge all my colleagues which have helped over a period of time.
Thank you very much, sir, for a wonderful session on recent advances in genomics and its applications in disease diagnosis and management. Now we have a few questions from the participants, sir. Just two, three questions. Rest we can take offline through the feedback link. First one, I would like to ask a question from Vallabh Srinivasanji, who is asking, is there any cure for diabetes and obesity?
Cure is has a very different. Cure means that you one time treatment, then you are cured of this, right? So I would say that there are yes and tried different types of transplantation, central transplantation, fecal transplantation. Right? As of now, I can only say that fecal transplantation is cure for diabetes. It's management of the disease, because we come. Uh,
Okay, sir. Thank you, sir. There is one more question. Is asthma solely genetic disease or caused due to environmental changes like allergies?
no, asthma is a complex disease.
Thank you, Chandak. I just want to leave.
Thank you,
Okay, thank
sir,
you.
Padmanabhan sir, for a wonderful session.
Thank you. Thanks a lot.
Thank you,
Yeah.
sir. Thank you, sir.
Yeah.
Thank you, sir. Bye, sir. Please. Sir, there is one more question. It's regarding asthma solely genetic disease or caused due to?
Yeah.
Yes, sir. Yes, sir.
Asthma, yeah. Asthma is also a complex disease. When it all started long back, it was thought to be because of allergy. But then subsequently, we also know that although there are allergens all around, it is only few individuals which give their response like and leading to asthma. Some right. So they become all right. There are, genetic factors like these interleukins in particular, which decide the T cell response, and that's how the asthma occurs. And there are genetic variants in these genes which show the susceptibility. But at the same time, environmental factors, like taking a specific kind of diet or keeping yourself healthy, these are also important factors. So it is also a category of complex diseases.
Thank you very much, sir. Any words from Shantanu Das, sir? Dr. Shantanu Das? Any questions?
Sir, no questions.
I have.
Okay. Yes, sir.
Sir, I have 3 questions for you. Hello. Sir, when we go for blood analysis for diabetes, should we do FPG, PPG, and HB1AC altogether, or HB1AC is enough to get the picture?
So as I said, for diagnosis, in the first time, you will just need a random blood. As I said, that's a because now there are specific criteria.
Yes, sir.
Go for fasting blood as well as postprandial blood glucose. HbA1c does not have an immediate implication as a diagnosis. It is more important for the follow treatment. Now, suppose you are diabetes and you are given a drug. You would not be measuring your glucose level every morning, evening like that. So if your glucose levels have been successfully kept low, then your glycosylated hemoglobin will be level which will be showing for last 3 to 4 months. So glycosylated hemoglobin has a value in the in determining the prognosis. So I don't think it is important that all 3 of them should be done together.
Yes. Second question, one of my colleagues' daughter has developed diabetes at the age of 9. Is it possible to go for gene sequencing now and hello?
Yes, sir. Please go ahead, sir.
Which disease? Diabetes.
One of my colleagues' daughter.
Which disease?
Hello. My colleague's daughter has developed diabetes at the age of 9. Now, is it necessary to go for gene sequencing and then go for treatment, or they already have started treatment now? That's okay.
So this is what I try to.
Hello. Yeah, correct. You told.
Sorry, your voice is breaking, sir. Yes.
Yes, sir.
So,
Yes, sir. Yes, sir. She is under treatment with me. Yes, sir.
yeah.
Hello.
Yeah, can you hear me?
I think there is a network issue.
Can you hear me?
No, sir. Now it is visible.
Yes, sir. Now it's visible.
Yes,
Yes,
sir.
sir.
So I'll switch off my video. So was saying that this individual can be either type 1 diabetes or can be MODY. Okay.
Oh, okay.
Now for MODY, if you go for gene sequence,
Yes, sir.
people manage
Oh, yes. Yes,
management is.
yes, sir.
Could you hear what I say?
Yes, sir. Partially I heard, but could you please repeat, sir?
So I'm saying that it will be good to go for MODY gene or if the
Good to go for gene sequencing.
uh,
Hello.
so you can try in Chennai. There is Dr. Mohan's Diabetes Center. They are there.
Yes, sir. Oh, okay. Okay. Thank you, sir. Hello. Yes, sir. Thank you very much for an exciting and very informative talk. On behalf of KST and all the
Sure.
participants, we thank you immensely, and we will be inviting you for our conferences in Bangalore, which we hope to organize maybe in December or January period. You are a very excellent speaker,
Okay.
sir. Thank you very much. Very kind of you for your time
Thank
and
you.
talk. Thank you very much. Thank you, sir.
Thank you very much. It was my pleasure also.